Can you hear me? Okay, good. So, good morning. Sorry, I'm just going to apologize in advance. You have to bear with my Singaporean accent this early. So, yeah, but I'm sure you all do well. So, okay. So, I'm going to talk about our new study regarding optimizing the balance of sample size and scan duration in resting fMRI studies. So, maybe I don't need to belabor this point at this conference, but we have seen success in using resting state fMRI to perform individual level predictions in things such as brain maturity and fluid intelligence. And individual level prediction is an obviously important task because we want to know how individual differences in brain network organization relates to phenotypic variation across participants. And it's an obviously important task for an important component for precision medicine. So in the current landscape of studies, there are many studies that tell us that larger sample sizes are important. So I'm not sure, maybe you know this one already. So this one tells us that we need thousands of individuals for replicability. And there are other studies that look at this in terms of prediction accuracy as well. So in this study by He in neuroimage, he compares the different performances of machine learning and deep learning algorithms while changing the sample size. And you can see this in this graph over here. So as he increases the sample size, we see that prediction performance actually increases. So it's beneficial for our prediction accuracy. So something that's less talked about is how much time do we need to put our participants in a scanner for? So here's some work by Feng in cerebral cortex. So he investigates the influence of fMRI time points while keeping sample size fixed. So if you look at this cognitive factor score, you see that prediction performance also increases if we increase the number of fMRI time points we use to perform our prediction. So then this brings about the question. So normally, I have a fixed budget. So I can either optimize my scan time, or I can optimize the number of participants I collect for my study. So which one should I do? So we try to give more detail and try and look into the factors that, which factor is actually more important for this. So let me give a bit more detail in the next slide. So basically, we've constructed FC matrices from different amounts of time, starting from 2 minutes to the full duration of the scan. And we use this as input features to predictive models to predict different non-imaging phenotypes. And we change the number of participants we use to train each predictive model. And we conducted this analysis in 2 datasets, the ABCD, or Adolescent Brain and Cognitive Development Study, and HCP, the Human Connectome Project. So let me just move on to the results really quickly. So okay, over here, I'm showing the results, the prediction results for the Human Connectome Project. And this is a cognitive factor score we derived in the Human Connectome Project. So on the x-axis here, we have the scan duration per participant. And on the y-axis here, we have the number of training subjects we use to train the prediction algorithm. So each combination of training subjects and scan time per participant has a brightness that corresponds with how well it was predicted. So let's first look at a fixed scan duration, for example. So if we have a fixed scan duration, for example, 34 minutes, and we increase the number of training participants we use to train our model, we can see that prediction accuracy increases. And likewise, if we flip this around, and we increase the number of the scan duration per participant while keeping the number of the sample size fixed, we can see our prediction performance increases as well. So we can zoom out into this full plot, and we see that both sample size and scan duration per participant helps to improve prediction performance. So let's see how this behavior replicates over 2 datasets. So over here, we have both datasets over here, and then we can see the results across both datasets. So over here, we additionally draw some lines to show that the areas where prediction accuracy is almost equal. And we can see that for both datasets, there are many different ways to get a specific prediction accuracy. So if I want a prediction accuracy of 0.4 correlation, for example, I can do it with 2 minutes and 1,800 subjects, or 18 minutes and 400 subjects, for example, right? So this still doesn't give us an indication of what is more important. So let's try to look at this in a different way. So over here, let's try and rethink this. So for example, so over here, we are going to keep prediction accuracy on the y-axis. And this time, we're going to plot the x-axis as total scan time, which is number of training participants multiplied by scan duration per participant. And we're looking at a cognitive factor score in the ABCD over here. So what we can see is that, amazingly, total scan duration can explain prediction accuracy really well. And over here in this plot, darker colors indicate larger training set sizes. And so dots of the same color have a larger scan duration as you go from left to right down this plot. Okay, so let's see how this replicates in the HCP. So just to give a brief reminder, in the ABCD, we scan our participants for up to 20 minutes, whereas in the HCP, we have a whopping 58 minutes. So how does this hold up? So we see, again, total scan duration explains prediction accuracy really well, but there's something strange going on. You see, at the end of the curves, at this points in black, we see that eventually, when you increase your scan duration, the amount of time you scan your participants, eventually there's a fall off. And you don't get so much utility from scanning your subjects even longer. So, but if we ignore this for now, and we try to model this first part of the equation using a logarithm curve, it seems like using the first 20 to 30 minutes might fit a logarithm curve pretty well. So that's what we did. So just to show our equation over here, we fit accuracy as A times the logarithm of total scan time plus B. And we estimate A and B from each phenotype that we looked at, and plot the normalized accuracy here on the y-axis. So the reason why we use normalized accuracy is because some phenotypes are predicted better than others. So this allows us to plot different phenotypes on the same axis. So I'm just going to show you the results for the cognition factor scores in the HCP and ABCD first. So we looked at different prediction algorithms, different accuracy metrics in these two data sets. We compared linear ridge regression, kernel ridge regression, Pearson's R, and coefficient determination, COD. So what we can see is that across data sets, across regression algorithms, and across accuracy metrics, total scan time can explain prediction accuracy pretty well. So we see this by how well these points fit to the logarithm curve shown in black over here. So okay, let's repeat this for different behaviors. So over here, we have 34 other behaviors spanning the areas of cognition, mental health, personality, emotion, well-being. And we see that if we do this again, we see that most of these behaviors fit to this logarithm curve pretty well. So this gives us a sense that scan time and sample size are quite interchangeable below 20 to 30 minutes. So I guess being scientists, we question this a little. Is this really always the case? So we looked into this into more detail. So again, what we're going to do here is going to keep prediction accuracy on the y-axis, but this time we're going to hold the total scan duration constant at 6,000 minutes. So we have 6 combinations over here. So moving from an n of 600 down to 100. So this means we increase our scan time from 10 to 60 minutes. So what we can see is that as we decrease our sample size, the prediction performance decreases. So this gives us an indication that sample size is more important than scan time. So let's see whether this holds up for the ABCD as well. So we looked at the average accuracy of 19 phenotypes, and we hold total scan duration constant at 4,000 minutes. And we see that as we change n from 1,000 to 200, prediction accuracy decreases as well. So this gives us an indication that sample size is more important than scan duration. So can we explain this in an equation? So fortunately for us, we have our co-author Tom Nichols, who came up with a model that helps us to explain this. So he has a closed form formula that describes accuracy as a function of scan time and sample size. So I'll just briefly break down this formula. So K0 here is the maximum possible accuracy. K1 is a term that is controlled by sample size, and K2 is a term controlled by total scan time. So using this equation, we can fit this to one of our phenotypes, and then we can see how well it does. Right? So let's look at this in the HCP. So this is spatial orientation, for example. So if you look at the curves in blue, this corresponds to Thomas' model, and you can see that actually it fits the data pretty well. So what this tells us is that because of the K1 over n term, sample size is always slightly more important than scan duration. And taking a step back, this agrees with the intuition that inter-individual variability can be captured by larger sample sizes in terms of brain features and phenotypic variation, but not just scan duration. All right, so now we have this handy model. So how can we use it? Can we use it to tell us how much time we need to scan, how many participants we need to collect? Okay, so let's come back to this slide, and then maybe we just look at this equation a bit further. So if we ignore K0 for now, the second half of the equation can be thought of the fraction of maximum accuracy we can achieve with a given sample size and scan duration per participant. So since we have 36 phenotypes that we looked at, we can try and average this across the different phenotypes that we have and create a reference for what we're going to get with these phenotypes. So what I mean can be seen over here. So on the x-axis, we have the participants, and on the y-axis, we have the scan time per participant. So every combination has a color that indicates the fraction of maximum accuracy you can achieve when you plug in this n and t, right? So here's the result. So if you look at this result, the black lines here correspond to places where the fraction of maximum accuracy was equal with the different combinations of scan time and sample size. So this gives us an indication that there are many different ways to get a specific prediction accuracy. And what's interesting is that it starts high on the left and dips as you go towards the right. And this is because scan time is cannot just be ignored. If you increase your scan time, you can collect fewer participants and get the same prediction accuracy as well. So however, since sample size is always more important, do we just ignore it? Do we just scan everyone for 1 minute then? So unfortunately, I don't think we can, because samples, the cost of increasing your sample size, the cost of increasing your scan time are not the same. So what do I mean? So let's look at this in an example. So let's say we have a fixed fMRI budget of 100K, and it costs $500 per hour of MRI scan. So in this plot over here, on the x-axis, we have the scan time per participant, and on the y-axis, we have the fraction of maximum accuracy that is afforded by our budget. So that this means as you go from left to right on the x-axis, we are increasing scan time, but decreasing the number of participants because we pay for more scan time. Next. Okay, so I'm going to show you the overhead cost corresponding to 5 different scenarios. So the first scenario is $0, which is a volunteer or a PhD student. Second, all the way to $5,000. And there's going to be a dot for each curve that corresponds to the optimal scanning time that achieves the best prediction accuracy. So we see as we increase our overhead cost per participant, the dots shift from left to right. And this is because the cost of getting a new participant is so much more than increasing your scan time. You should just scan your subjects for longer. All right, so on to the results. So our results show that there's capacity for increased flexibility in study design. The sample size and scan duration are quite interchangeable below 30 minutes of scan time. But also do note that a good sampling of population variability requires a large N. And we also show that economic considerations play a large role in study design. Typically, increasing your subjects may cost much more if you need them to go through a battery of other tests, imaging modalities as part of the study as well. So some other factors that you might want to consider when looking at a study budget. So firstly is the population you're looking at. So some populations like children might need practice scans, or you need to compensate for their movement by having longer scans. And besides this, some other consideration is also the number of sites you're using. Say you can't collect everything on a single site. Having multiple sites will also increase the cost because you need more RAs or PIs to manage the different sites. Okay, so I'd like to thank our funding and support, and our lab members as well. And thank you. So yeah, you can find this preprint of our study in the QR code below. And if you'd like to talk more, I'll be at poster 13A this afternoon. So thank you.
Thank you, Leon. Let's leave for those who are late, we're going to leave the questions till the end. So the next speaker